Alright, welcome back to another video. We're going to take a look at the immune system in this lecture series. So the immune system is going to largely consist of different types of immune cells, so our different leukocytes, our white blood cells, and this system will be closely related to the lymphatic system, and we'll see some of the same cells and some of the same terms that we saw in the lymphatic system, but we'll also introduce some new terms. So before we get started, think for a second about what helps to comprise the immune system. So we already said some different types of cells. What else might you think will be part of our immune system? So think about that as we get into the lecture. So the immune system is going to be one of our bodily systems that has different cells and organs that is going to, it's designed to protect us from foreign pathogens. So anytime we say pathogen, that is just some type of foreign. It can be a bacteria, it can be a virus, it can be a parasite, it can be anything that is not supposed to be in our bodies. We can take this immune system very broadly and then divide it down into two general classes. You can have innate and you can have adaptive. So if something is innate to you, you are born with it. You do not have to work to develop it. So we have different types of innate immune system features that are part of our body. So we have our skin, so barrier, mucous membranes, and then we have different types of cells that are going to be part of our innate immune system. And the idea here is that the innate immune system is very non-specific. It doesn't, sometimes you'll hear in the literature the word discriminate. It doesn't discriminate against one type of pathogen or another. It just knows that something is foreign, it is not supposed to be here, and so it will act to destroy and get rid of that pathogen. So it's very non-specific in that manner. You can contrast that with our other type of immune system, the adaptive immune system. This one, unlike the innate, which we're born with, the adaptive immune system we have to develop over time, and we develop it by encountering different types of foreign pathogens, foreign antigens. So whereas the innate immune system was very non-specific, it did not discriminate against one type of pathogen or antigen from another, the adaptive immune system is going to discriminate from one type of pathogen to another. So it is very specific. It targets a specific antigen or a specific pathogen. It does this primarily through two types of leukocytes. Remember, leukocyte is a white blood cell. That specific type of leukocyte is going to be our lymphocyte. And then we said that you can further divide lymphocytes into two more subcategories of T lymphocytes and B lymphocytes, so T cells and B cells. And we'll see how these T cells and B cells are incorporated into two types of immunity that we'll call cell-mediated or cellular immunity, and then humoral immunity. So looking at this picture, you can see up at the top, you can see a very broad stem cell. So a stem cell is going to be this undifferentiated cell that has the ability to divide and differentiate into all different types of cells. So we start here. We will go to platelets, we can go to red blood cells, and then we can go to a lot of these other ones, which are all types of leukocytes or white blood cells. So we have neutrophils, basophils, eosinophils, monocytes, and then we have our lymphocytes over here with our T and B cells. We'll spend a lot of time talking about T and B cells, and then ultimately plasma cells down here when we get to the cell-mediated and the humoral components of our immunity. But just appreciate this chart and see how everything is flowing from the one common it's called a multipotential hematopoietic stem cell or hemocytoblast. It has the potential to divide into multiple different cells. So that's why we call it that. And then hematopoietic, this is referring to the creation of blood cells. So all these are different types of blood cells. These are white blood cells, these are red blood cells, and then these are our platelets. So our first type of immune defense that we're going to look at is our barrier defense. Okay, we can classify our barrier defense as being innate in that we don't have to develop it. We just have our barriers. We can classify it as being very non-specific because we do not, it does not discriminate against one type of pathogen or another, and then we can classify it again as our first line of defense. So this makes sense if you think about your skin, or you think about a mucous membrane lining your oral cavity or your respiratory tract. This is going to be the very first thing that a virus or a bacteria will encounter before it gets deeper into your body. So again, innate, it is very non-specific, and it is our first line of defense. It's one of our first lines of defense. So some different types of barriers we have. We have acid in our stomach, so our gastric juices. This will serve, one, for digestion, to help digest and break down different things we ingest, but that doesn't really have anything to do with the immune aspect. The immune aspect is that this acid has a very low pH, so a pH, if you know, anywhere from maybe 2 to 3, 2, 3, 4. And this low pH, this high acidity, helps to inhibit the growth and replication of different types of bad bacteria that we may ingest or that may otherwise get into our gut. We have antimicrobial protein, so lysozyme. Lysozyme is a protein that is found in our saliva that has antimicrobial properties that will break down different types of bacteria. We have mucus in our respiratory tract and our gastrointestinal tract that is essentially a thick substance. You can picture mucus, a very thick substance that is also going to act as a barrier, but it will act as a barrier in the sense that it will trap and kind of keep viruses and bacterium lodged within it. And then we can have something called, and we'll talk about when we get to the respiratory system, a mucociliary escalator. And so this is due to the presence of small cilia, which are these little hair-like projections on our cells in our respiratory tract that beat rhythmically, and it will act to move this mucus upward toward the pharynx and ultimately to the oral cavity, and we can either get rid of that through spitting that out, or we can choose to swallow that into our gastrointestinal tract. And then lastly, another barrier defense is bacteria. So I just said that our immune system is going to act to prevent viruses and bacteria from taking root and causing problems. So how does it make sense that bacteria are now going to be part of our immune system in the sense that they will help us? So take a second and think about how are bacteria actually going to be beneficial to us. So maybe you paused the video, maybe you thought about it for a second. The idea here is that bacteria will be present in our gastrointestinal tract, and when they're present in our gastrointestinal tract normally, we'll call them our normal flora, our bacterial flora, normal gut microbe, okay? And what these bacteria do is they act to out-compete for nutrients that would prevent bad bacteria from taking root and beginning to develop and grow. So essentially, they're there to take nutrients and compete for the same nutrients that bad bacteria, that we would classify as just very generically bad, that they would also want. So they're essentially in there to prevent others from taking root. Now, when you take an antibiotic, you generally try to take a very specific antibiotic that is designed to target a very specific microbe that you have as your current infection. So you do not want to give, you can initially, but you don't want to give broad spectrum antibiotics for a prolonged period of time, because these are antibiotics that target roughly, in a lay sense, bacteria in general. So whenever you target bacteria in general, you're also targeting those good bacteria in your gut, so you can wipe those out. And when you wipe those out and get rid of those, now you've opened up for the opportunity for bad bacteria to take root and begin to develop in your body. So you'll see this in the clinic. You will always get a culture first when somebody has an infection. Before you start antibiotics, before you do anything like that, you get a culture. So you take part of that infection, wherever it is. If it's in the blood, you'll get a blood sample. Wherever it happens to be, you get a culture first. You send it to the lab, they grow it up, and they test different antibiotics to see which one is the most specific for that infection, and then they will relay that, and you will receive that very specific antibiotic as opposed to broad spectrum antibiotics. So here's just a picture that is showing our different immune defenses. So you have our saliva with our lysozymes, skin, stomach acid, mucus linings, tears, as well as those good, normal, healthy bacteria that we have in our gut that are going to outcompete for the nutrients that bad bacteria would also try to take. So the next part of our innate immune system are going to be cells and chemicals. So again, keep in mind, this is still innate, so this is very non-specific. These are things that we are born with. We don't have to act to develop these. They are just present in us. These are going to be classified as second line of defense, so our cells and chemicals. We have a class of cells called phagocytes. And so what phagocytes are, these are cells that undergo and perform phagocytosis. So what is phagocytosis? Well, phagocytosis is generic cell eating. So there is a virus, there is some type of foreign particle, there is an old worn out dead cell or old cell debris. Phagocytosis is the process of a cell bringing that in, so whether it's a virus or bacteria or old cell part, bringing that into itself, combining it with a lysozyme, and then helping to break that particle or that virus or that bacterium down to where it is no longer a problem. So I'll show you a picture of what phagocytosis looks like in the next slide or two. And then we also have something called a natural killer cell. So a natural killer cell is a type of lymphocyte. And what they are is they are immune cells, and they can target abnormal cells or foreign cells. So in the sense of abnormal cells, we're talking about our normal healthy cells that have now mutated and are in the process of possibly becoming a cancer. So these cells can recognize these and check them and feel them to see if they're abnormal, and if they happen to be abnormal, they can act on them and destroy them. So this is a picture of a Kupffer cell. And so a Kupffer cell is a specific type of phagocyte or a macrophage that is going to be present in only our liver. So we have, if I go back for a slide, we have macrophages. So we have these macrophages in different parts of the body that are specific to just that part, but they perform roughly the same function no matter what area they're in. So we'll look at the liver here with our Kupffer cells. They line these sinusoids, and when we get to the digestive system, we'll discuss what sinusoids are. They line these sinusoids, and any blood and fluid that is floating in the sinusoid that happens to have a foreign bacteria or virus or something like that, these Kupffer cells, these macrophages, can engulf that and take care of it. We have these small little green structures here. This is an actual image, a scanning electron microscope of what these green cells are. These are alveolar macrophages. So if these are alveolar macrophages, where do you think these are found? Well, hopefully you're thinking the lungs because of the alveoli. But these are specific to the lung, and they perform a very similar function. Sometimes they're referred to as dust cells. But anything that we inhale in the air that happens to make its way down to the alveoli can be processed and engulfed and taken care of by these alveolar macrophages. This is a microglia cell. And so these are common and found in the nervous system, in the central nervous system, and they perform a similar function. They are the macrophage phagocytic cell of the nervous system, just like you have the Kupffer cells in the liver, and here you have the alveolar macrophages in the lung. So this is the general process of phagocytosis. So here you have a bacterium. So this is our foreign pathogen. Here you have a macrophage, this large cell here. It will, via endocytosis, bring in this foreign bacteria, package it in this vesicle here called a phagosome. Okay, when the phagosome and a lysosome fuse, they will form a phagolysosome. And what the lysosome has, if you think back to AMP1, they contain digestive enzymes. So when the lysosome fuses with the phagosome, the lysosome contributes its digestive and lysosomal enzymes to break down this bacteria. So here we are now, it's broken down, and then these will be expelled via exocytosis into the extracellular space. But it's this idea of bringing in something foreign, combining it with a lysosome and ultimately breaking it down so that it is no longer an issue. So one of our last innate immune defenses is general inflammation. So we're all relatively familiar with what inflammation is. Inflammation is going to be a series of symptoms that we see after some type of tissue injury. Now, this tissue injury can be an infection, but it could also just be some type of generic tissue injury. So maybe some type of mechanical injury if you fell and scraped your leg or your arm, if you twisted an ankle, if you happen to break your bone, you'll see some of these signs of inflammation. So there are generally 5 typical signs, or sometimes they're called cardinal signs of inflammation. And so there is redness, which is called rubor. There is heat, which is called calor. There is swelling, called tumor. Pain, which is dolor. And then this last one is sometimes contributed in and sometimes it's not, but a general loss of function. So if you think about, let's just take the example of twisting an ankle. Now this isn't an infection per se, but it's still something that leads to inflammation. So think about when you roll your ankle. If you roll or twist your ankle, you generally have it be a little red, and this red can be to the increased blood flow that occurs after a sprained or twisted ankle. You have heat, and the heat is also, it's due to the presence of some blood. You have swelling. Swelling can also be due to the blood, and it's also due to the dilation of blood vessels in that area due to the injury. You obviously have pain when something like this happens, and generally you can't use that ankle as well. It's very painful and sore, and so you can't perform the same functions on it as you normally would. So this graph was one that I was shown in school as well. It's a very good one to help you remember the different signs of inflammation. So you see heat here with the fire. You see redness as this man's face is red. Swelling here. You can see pain here, and then a general loss of function. He's unable to perform his normal function. So think about it in terms of, if you twisted an ankle, or if you were to have an insect bite or some other type of infection, what would you likely see? So in this image, what do we see here? So obviously we see somebody's foot and their toes that have undergone some type of tissue injury. Now this doesn't necessarily look like a mechanical injury. This looks like perhaps some type of infection. But you can see here it looks very red. You can see that the toes look generally swollen. This does look like it would be warm to the touch. They probably do not have as much function with this as they generally would, and this is probably pretty painful, you can imagine. So this may be due to some type of infection that has entered into this foot area. So the process of inflammation is going to be a series of steps that are occurring from the time the tissue is injured until times afterwards as the healing process begins. So what we'll start with is tissue injury. Okay, so we injure our tissue, so we mechanically damage it, we have some type of infection, and what happens is the cells, the immune cells, and then the endothelial cells of the general area will release different chemicals. Okay, so we have histamine, leukotrienes, and prostaglandins. So what these different chemicals are going to do, or they're going to result in different things. So histamine results in some vasodilation. Leukotrienes and different ones can recruit more cells to the area, and then prostaglandins are ultimately what result in the pain associated with inflammation. So different medications like Advil and ibuprofen, things like that. Advil is ibuprofen. It's just different names for it. They inhibit prostaglandin synthesis. And so, actually, I believe it's Tylenol that inhibits prostaglandin synthesis. But when you inhibit prostaglandins, you inhibit ultimately the pain sensation that you feel. So that's how Advil and Tylenol and acetaminophen, things like that, that's how they act to reduce the pain you associate with inflammation. So The next step we'll have after tissue injury is we have vasodilation. And again, it's due to some of these chemicals that are being released that result in the increasing of the vessel diameter. So with vasodilation, we now get the swelling that's associated with inflammation, as well as the redness, because now we have more blood in that area, so it appears swollen, and that blood contributes to the red color. Now, with this vasodilation, it allows for more blood to be present at the area of tissue injury. So if you have more blood at the area, you hopefully have more immune cells at the area, and that's really one of the important things with vasodilation is to increase the availability of lymphocytes and different types of leukocytes to that area that can then act to take care of any foreign pathogens that may have entered during the injury process. Now, number 3 is something that we're going to call increased vascular permeability. So this can occur due to the presence of chemicals that will cause the vasodilation, and when this happens, the different cells lining the blood vessels tend to move a little bit apart. They move further apart, and so when this happens, it allows for cells and blood to leak out of the vessels and enter the tissue space, the interstitial space. So this is what results in some of the swelling, or what we call edema, associated with inflammation. Now the last step is going to be the recruitment of phagocytes. So obviously, if we have an injury and we have inflammation, we need to be able to bring in immune cells from the surrounding area, and so this is going to be the recruitment of phagocytes. So neutrophils and macrophages, which are both phagocytic cells, they'll arrive at the area and ultimately act to break down and endocytose foreign particles and foreign pathogens that will then be destroyed. Now, the way these cells leave the vessel, so they're in the blood vessel, and they have to get out of the blood vessel and go into the interstitial space around the cells and tissues where the injury or the pathogens are. Now, they are recruited to the area via positive chemotaxis. So that means that certain cells will release chemicals, and these phagocytes respond positively to those chemicals and go toward them. Okay. Now, this term diapedesis, this has to do with how the cell is leaving the vessel. So the cells of the vessel will move apart slightly, and these cells will essentially squeeze through and push their way through these cells, and then they can enter into the tissue space to perform phagocytosis. So here you can see our general series of steps. So we have a tissue injury first. We have increased vessel diameter, so vasodilation, and increased vascular permeability here to allow things to leave through. So here you can see diapedesis occurring as these cells are squeezing through this blood vessel. And then here we have the recruitment of phagocytes to the area, and they will be phagocytosing any foreign bacteria and cellular debris that may be present, and ultimately will result in the healing of this injury. Now, this picture, there are lots of things here. There's a whole series of processes that occur as white blood cells, leukocytes, are leaving the blood. So all this up here, this is all blood, this red up here. These are the cells lining the blood vessel. And here you can see a leukocyte undergoing diapedesis as it squeezes its way through two adjacent endothelial cells that have now moved a bit apart. So there's this whole thing of rolling, activation, adhesion as it adheres to the endothelial cells, and then it squeezes through. And there's a whole series of molecules that act with this. But the idea here is that it is just squeezing through these vessels. Now it has entered this space, and it can perform its functions as it relates to the tissue injury. Now, we have one more innate immune defense that we're going to talk about, and these are going to be antimicrobial proteins, and then we'll also have fever as well. So one antimicrobial protein is going to be an interferon. So what an interferon is it is a chemical or a protein that is released by a cell that has been infected with a virus. So as a cell is infected with a virus, we're going to essentially denote that cell as being gone. It's dead. It will not be revived. It is too far gone. But that cell is still important because it's going to send signals to other surrounding cells, adjacent cells, to secrete antiviral proteins so that they themselves can be essentially safe and secure and protected from the virus that it was infected with. So it's essentially we're sacrificing one cell. It sends out these interferons that stimulate the cells surrounding them to create lots and lots of these antiviral proteins to prevent that same virus from entering it and multiplying and resulting in the same effect. Okay. Now, the second one, the complement system. This complement system is a long series of proteins that undergo changes throughout this process. One protein results in a change to the next protein, and it results in the change to the next. And it's this whole process of cleaving pieces off of a protein and changing them. But what you result in is you get this final protein structure that will target a bacteria. It essentially puts a hole in it, punches a hole, allows lots of water to rush into the cell or the bacterium, and that results in the cell lysing or swelling and then ultimately bursting. So I'll show you a picture of that. Here you can see interferons that we talked about. This cell on the left has been infected with a virus, same with this one up here. It secretes the interferon to this adjacent cell, or these cells down here, and as that occurs, it signals the cell to create lots of antiviral proteins to prevent the replication of the virus in those adjacent cells. Here is the complement system. Now, you start here with a pathogen. We binded antibodies to this antigen, which is on the surface of the pathogen. And when we have certain antibodies there, we can get complement activation. So when we get complement activation now, we get this whole series of steps leading all the way down. And what you end up with is you get this final protein structure here, this membrane attack complex is what it's sometimes referred to, the MAC, the membrane attack complex. What you can see is it punches a large hole in it, in this bacteria, this pathogen. When it does that, it allows water to quickly rush into the cell here, and when this happens, water rushes in, the cell begins to swell, and ultimately results in the lysis of the pathogen. Okay? So you don't have to worry about the individual proteins and what converts what protein to the next one and so on, but understand that you start with a pathogen that is bound with antibodies. These antibody complex can stimulate the activation of our complement system. You get this series of proteins working all the way down, and ultimately you get the membrane attack complex punching a hole in the cell, resulting in water influx and ultimately the cell lysing. So the last one we'll talk about here is fever. Fever is going to be a beneficial response to some type of infection or something like that. And what it is it's an elevated temperature. So if you have a fever, please don't say that you have a temperature, or you'll hear people say, "Oh yeah, I've got a temperature." Well, yeah, everybody has a temperature, right? If you don't have a temperature, you're probably dead. Okay, so say you have a fever, you have an increased temperature, which is a fever, and it's a result of chemicals called pyrogens being released from leukocytes, different types of white blood cells, and macrophages that work on the hypothalamus in the brain to increase our internal thermostat, essentially reset our temperature to be higher. And what this higher temperature does, this higher body temperature does, is it inhibits viral and bacterial replication in our cells. So if we increase the temperature and prevent viruses and bacteria from replicating, now we're going to prevent them from hopefully becoming a very serious infection and spreading even further. Now, your temperature, your fever, it can become too high. Okay, so you'll see on the unit, if you're already a nurse, you probably know this. If you're not, it's okay. You'll learn it in nursing school. But different units and different clinical settings have different thresholds of when you begin to treat a fever. A somewhat higher body temperature, this fever, it's good. It's normal. It's healthy. It's protective. Now, when you get too high, when you begin to get it 100 and 200, 300, 4 and above, this can become really dangerous. Now, our internal body temperature is normally 98.6, but like I said, when you really get 102, 103 and above, this becomes too high, and you begin to get the denaturing of proteins and enzymes, so they become inactive. You can cause things to die and get cellular death, and so you can get febrile seizures, in which you have a seizure in response to an increased fever or increased body temperature. And so there is a threshold that a fever can now stop being beneficial and actually become harmful. So what fever does is it increases cellular metabolism. Okay, so our cells become more metabolically active. It amplifies effects of the different chemicals that we've talked about, and it increases the difficulty for bacterial replication. So it essentially makes bacteria replication and the spreading of the bacteria more difficult. And this is a healthy protective thing. So here you can see this general process. You have a macrophage that engulfs via phagocytosis a bacterium. As it does this, it releases cytokines, or these chemicals that travel to the hypothalamus in the brain. When you stimulate the hypothalamus in the brain, you will increase your body temperature to result in a fever. So your internal thermostat is increased, and this increases the body temperature, which hopefully inhibits further bacterial replication. So we'll stop here, and we'll pick up with part 2 with our adaptive immune system.